Hello, my name is Jacqueline Chaparro, and I'm a research scientist at Colorado State University's Department of Horticulture and Landscape Architecture. In my work, I study the ionome, or the elemental composition of organisms. In a food context, ionomics, or the study of the ionome, includes the study of mineral, nutrients, and trace element composition of food that can have important implications for human and planetary health. But before we get into some of the applications of ionomics for the study of food, let's start by talking about what elements are. Elements are the building blocks of all things that make up our universe. You may recall from your chemistry class the periodic table of elements, which displays the full array of known chemical elements grouped by their similar chemical properties. Each element has unique properties, from helping our bodies to make energy, to building bones, fighting infections, and supporting brain function. Some of these elements are natural components of food, such as carbon and phosphorus, which are found in all plants and animals, or zinc and iron, which are essential minerals for human and plant health that can be found in both plant and animal foods. These elements are involved in critical functions for human health, with imbalances leading to deficiencies or toxicities. For example, zinc is involved in many physiological functions, including immune support, protein synthesis, and cognitive function. Low iron levels can lead to iron deficiency anemia. Certain elements, such as cadmium and arsenic, can contaminate our foods through environmental sources. When consumed, these trace elements can be toxic to humans. Understanding the elemental composition of our foods is important to help us get more of the nutrients we need from food and to avoid contaminants that can be bad for our health. Elements are also essential to the functioning of ecosystems and agricultural processes. Many minerals play a fundamental role in crop growth, development, and quality, as well as in soil health. Ultimately, ionomics unlocks clues about how elements shape the world around us. In nature, elements are incredibly dynamic, frequently moving within and between organisms and changing chemical form based on the functions they are performing. Oftentimes, the balance and function of different elements within an organism is tightly regulated by the organism's genome. However, the elemental composition of foods can be influenced by a range of factors in the environment, such as the soil and chemical composition, environmental stressors, and interactions with other biomolecular compounds, such as proteins and metabolites. Think of the ionome as a massive social network of different elements constantly interacting with the genetic, physical, and chemical environment. The most commonly used instrumentation to measure the ionome is inductively coupled plasma mass spectrometry. This instrument helps capture the snapshot of the elemental network of a given food. Food samples first need to be digested into their elemental forms. This typically involves the use of strong acids such as nitric acid, often combined with heat and microwave-assisted techniques to ensure complete breakdown of the samples. By reducing the sample to its basic elements, digestion allows for accurate quantification of elemental concentrations. The elements are then ionized and undergo a series of steps that ultimately allow us to quantify the concentration of different elements of importance within the food. One of the biggest benefits of the application of ionomics in the lab is that we can measure many elements at once in a relatively fast way to get a more robust picture of which elements, good, bad, or neutral, are in a food. Understanding the comprehensive profile of food is important to understand the interactome, or the complex network interactions between biomolecules in ecosystems, our diets, and in our bodies, such as the processes that influence nutrient bioavailability. For example, iron deficiency is one of the leading causes of anemia worldwide, leading to fatigue, weakness, and poor immune function. But the problem isn't just about eating iron-rich foods, it's about how much of it we can absorb from these foods. And this depends on molecular interactions. For example, zinc and iron interact with one another in the body with regards to absorption. High levels of one of these elements can interfere with the absorption of the other. Likewise, calcium, magnesium, and zinc may compete for absorption in the intestine, a phenomenon known as nutrient antagonism. Imagine such interactions for all the elements, and then further expanding these interactions between all the tens of thousands of biomolecules in the foods that make up our diets. While we know that elements are important for the health of people and ecosystems, we don't always know how much we're getting or how efficiently we're absorbing them. This is where the importance of measuring elements in food through ionomics comes in. Advancements in foodomics and ionomics are helping us measure elements more effectively than ever before. By applying ionomics, we can understand the levels of the full range of elements in the foods that we eat, allowing us to track element levels across different food systems in different regions and farming practices, unlocking new insights into food quality and health. Sometimes it is necessary to understand the different forms of an element that are present in food. The application of ionomics to food enables this. The toxicity and behavior of an element in biological and environmental systems depends on its specific chemical form. For example, arsenic exists in different forms depending on its source and chemical structure. And there are more than 20 arsenic compounds identified in nature and biological systems. Some of these, such as inorganic arsenic, arsenite, and arsenate, are highly toxic and classified as human carcinogens. On the other hand, organic forms such as arsenobadene, typically found in marine organisms, are relatively non-toxic. To determine the specific forms of arsenic in a food, specialized techniques for speciation analysis are required. Without this analysis, we risk overlooking harmful species such as toxic inorganic arsenic while misjudging safer organic forms like arsenobadene, leading to incomplete and inaccurate evaluations of the arsenic content in food. In addition to understanding the mineral nutrient content of foods and detecting the presence of potentially toxic trace elements, ionomics can be used to help understand how plants adapt to a changing environment to guide agricultural and breeding strategies. Ions have been identified as important players in plant stress biology, and ionomics can be paired with other omic approaches to support environmental monitoring by detecting changes and patterns in response to different stressors, such as those posed by climate change. Ionomics can also help identify potential biomarkers of plant resilience and track changes in food composition over time in response to different interventions. This becomes increasingly important as we learn more about the negative impacts of rising atmospheric carbon dioxide levels on the elemental composition of important staple crops, such as rice and wheat. It has been estimated that this climate-mediated nutrient dilution could result in a 5 to 15% decline in mineral concentration of food worldwide. With zinc and iron deficiency already contributing to millions of deaths annually, it's critical to more closely monitor the mineral composition of the world's food supply and identify strategies for improving nutrient density and food quality through more targeted and responsible agricultural practices. Ionomics is also revealing the incredible biochemical variability of different food species and cultivars. For example, a study of 60 distinct potato genotypes used ionomics to assess variation in mineral nutrients in both cooked and raw potatoes. The analysis of 26 elements revealed significant variation in potato mineral content within both genotype and market class, including key nutrients such as iron, zinc, copper, and calcium. The combination of low anti-nutrient factors and potentially complementary nutrients in some varieties of potato, such as ascorbic acid, which enhances iron absorption, makes potatoes an important food crop for improving nutritional intake of essential minerals. Identifying species of potatoes with higher mineral density and breeding for these types of nutrient traits is an important strategy for improving food quality and fighting malnutrition worldwide. The timing of harvest is also important when evaluating the mineral density of foods. For example, ionomics was used to analyze the mineral composition of microgreens compared to their mature counterparts. It was found that copper was 13.7-fold higher in broccoli microgreens than mature broccoli, and red cabbage microgreens were 2.4-fold higher in iron, 9.1-fold higher in copper, and 3.8-fold higher in zinc than their mature counterparts. Interestingly, some greens like pea greens were found to have lower levels of key minerals such as magnesium and calcium compared to their mature counterparts. The variability of mineral and trace element concentrations is also heavily impacted by environment and agricultural practices. Soil element composition plays a major role in determining the elemental composition of crops. Typically, farming practices such as tilling, fertilization, irrigation, and crop rotation can either enhance or deplete elemental composition of crops. Other ecological and agricultural factors can influence the absorption and accumulation of toxic elements in certain plants and animal species that may be harmful to health. Elements play monumental roles in ecosystems, food quality, and our health. But how can we know if our food has the right balance of these different components? The answer lies in measuring them through ionomics, because understanding the elemental component of our food doesn't just tell us what's on our plate, it tells us the potential of how that food is nourishing our ecosystem and our bodies. This knowledge can be leveraged to inform evidence-based agricultural practices, food policy, and even personalized nutrition. So the next time you pick up a piece of fruit or any meal, remember that food quality extends to the elements that are hidden inside. By measuring them, we open the door to healthier, safer, and more sustainable food systems. As Ionomics expands and enhances our ability to rapidly map the elemental composition of foods, we will learn more about the patterns of mineral and trace element composition across the world's food supply. This knowledge will reveal how the presence of nutritive and potentially harmful compounds vary with different food system attributes. Paired with other foodomics technologies and complemented by plant genomics, Ionomics is enabling us to better understand interactions of crops with their environment and identify new strategies for improving the nutritional quality and climate resilience of foods around the world. Understanding these interactions within ecosystems, foods, diets, and our bodies is key to developing solutions for improving human and planetary health.